So lots of you GCSE students in year 11 right now have mocks coming up. So I thought we would do a paper one walkthrough. So we're going to go through the whole paper together and we're going to answer all of the questions, and hopefully this will help you with revision for any paper one questions that you've got coming up soon. So let's go. We've got a root is a plant organ. Plant roots contain many different types of tissue. What is a tissue? So all they're looking for here is the definition of a tissue. So we can say a tissue is a group of the same or similar cells. And we can leave it there because it's only one mark. Or as an alternative definition, if you don't convey the idea that they are the same or similar cells, you could get the mark for saying a group of cells with the same or similar or with a specific function. I tend to just say both. I say a group of similar cells with a specific function, and then you're going to guarantee yourself the 1 mark. Tissue in the tip of a plant root contains stem cells. Stem cells can differentiate into any type of cell. Name the type of tissue in plants that contains stem cells. The answer here they're looking for is meristem or meristem tissue. So meristem tissue in plants contains stem cells that can differentiate or specialize into any type of cell. So that's just an easy 1 marker. Another easy 1 marker. Aspirin is a painkiller. Which plant does aspirin originate from? Now, aspirin comes from the willow tree, or you can just say willow. Another drug that you should know, for example, is the heart drug digitalis, comes from foxgloves, or penicillin comes from penicillium mold, which is a fungus. But yeah, for this, it's just 1 mark. Again, it's something we have to learn. Aspirin comes originally from willow trees or willows. Scientists have extracted chemical A from a deadly nightshade plant. Chemical A can be used as a painkiller, and table 1 gives us information about where chemical A is found. The scientists usually extract chemical A from berries. Suggest 1 reason why berries are used instead of leaves or roots. Now, there's lots of alternative ways to get this mark. For example, berries are easier to access, or berries are very easy to pick, and berries are easier to extract the chemical from, chemical A, or taking berries is less likely to harm the plant. The command word here, by the way, is suggest. Now, when you see that command word, it means you haven't learned the answer to this, but you should be able to apply your scientific knowledge and a bit of common sense to figure out a reason. So imagine for yourself that you're trying to extract this chemical. It's going to be far easier to collect the berries, pick the berries. It's going to cause less harm to the plant. You could say things like, taking leaves might damage the plant, taking the roots might damage the plant or might kill the plant. You could even suggest, like if you look at the table, you can see that there's a lower mass of chemical A in the berries compared to the leaves or the roots. So you could even suggest that maybe because there's a lower mass of chemical A in the berries, maybe it's going to be less harmful or less toxic, so it's going to be safer. But I think the most obvious thing really thinking about berries is taking berries is less likely to damage or harm or kill the plant. If you're taking all of the leaves or the roots, it is more likely to damage or kill the plant. But remember, suggest, there's going to be loads of answers on the mark scheme. It just has to be a sensible suggestion that makes sense from the information that you've been given. Okay, maths question then. The deadly nightshade plant has chlorosis, yellowing leaves. The mass of chemical A found in the leaves of the plant is 60% of the mass shown in table 1. We need to calculate the mass of chemical A in 200 grams of leaves with chlorosis, and we need to give our answers in milligrams. So we're going to get 4 marks for doing this question. So it's quite a lot. Okay, let's underline the key information. So the mass of chemical A found in the leaves of the plant is 60% of the mass shown in table 1. Now this is because, remember, this plant in particular has chlorosis, so it's obviously not producing as much chemical A. So we know we're looking at the leaves, so let's just highlight that in the table. So the leaves normally have 1.2 grams of chemical A, they have 1.2 grams of chemical A in 100 grams of the plant tissue. But we know that in this plant with chlorosis, the mass of chemical A is only 60% of that shown in the table. So the first thing we need to do is find 60% of 1.2. Now we could do that by taking 1.2, dividing it by 100 to find 1%, and then multiplying it by 60 to find 60%. Or if we know how to work out percentages more quickly, we could do 1.2 times 0.6 to find 60% of 1.2. And that would give us a mass of 0.72. So that's the mass of chemical A in this particular plant in the leaves, because it's only 60% of the mass shown in the table. Now, the next thing we have to do, the table told us the mass of chemical A in 100 grams of plant tissue. We want to calculate the mass of chemical A in 200 grams of plant tissue. So the next step, and the second marking point, is to times this by 2. We're going to double it because we want to know the mass in 200 grams, not just 100 grams. So we're going to have to double that value, and we're going to get 1.44. Now, we also need to give our answer in milligrams, not grams. So to convert from grams to milligrams, we're going to times by 1,000 because there are 1,000 milligrams in 1 gram. Okay, so if you write it like that, 1 gram equals 1,000 milligrams. You can see you're going to have to multiply your answer by 100. So by 1,000, sorry. So we're going to get the third marking point by multiplying it by 1,000. And we're going to get the fourth marking point for getting the final answer of 1,440 milligrams, okay? So let's go through the mark points again. If you work out what 60% of 1.2 is, you're going to get marking point 1. If you times that by 2 to find the mass in 200 grams, you're going to get marking point 2. If you multiply that by 1,000, so you're converting it to milligrams, you're going to get marking point 3. And if you get the final correct answer of 1,440 milligrams, you're going to get marking point 4. Okay? Show you're working, because if you don't get the final answer correct, you can still get marks along the way for all of your working out. 1.6, suggest one reason why the leaves of the deadly nightshade plant might have chlorosis. Now, again, this is a suggest. There's going to be loads of answers on the mark scheme. You've probably learnt that chlorosis is caused by lack of chlorophyll. But you might also have learnt that a lack of chlorophyll can be caused by a lack of magnesium or magnesium ions, okay? So if the plant hasn't got enough magnesium, it can't make enough chlorophyll. So lack of magnesium or lack of chlorophyll. They'll even allow lack of chloroplasts as an alternative. They'll allow lots of answers. They'll allow a lack of other ions, like a lack of nitrate ions, a lack of potassium ions, a lack of zinc ions. What they won't allow is lack of nutrients, because nutrients is too vague, okay? But if you've revised this and you've learnt this in school, you have probably learnt specifically that magnesium ions are needed to make chlorophyll. So you're probably going to go for lack of magnesium or lack of chlorophyll. Obviously, then the plant is not going to look green without enough chlorophyll. But yeah, loads of other things. They even accepted things like maybe the soil was too acidic, maybe the soil was too alkaline, maybe there was a lack of light Again, it's because the command word is suggest. So they've got to accept any potential answer that could have caused yellowing of those leaves. Chemical A has not been tested in a large scale clinical trial in the UK. It's important for drugs to be tested in clinical trials before the drugs are approved for use by the public. Give 2 reasons why. So why do we need to carry out clinical trials on any drug? We can say things like to check for toxicity. We can say to check dosage or find the best dosage that we should be using. We can say to check for efficacy, which basically means, does the drug actually work? Does the drug do what it's intended to do successfully? Now, alternative marking points. If you don't say check for toxicity, you can be able to say check it's safe. That was accepted. Or check it's not poisonous. That was accepted as well. Check it's not dangerous was also accepted. Instead of to check for dosage, they accepted things like check how much of the drug is needed. To check for efficacy, they accepted things like check if the drug actually works. They'll also allow check if the drug interacts with other drugs. Because you would need to know that, right? In case the patient is taking other medication, you'd need to check that the drug did not interfere or react or interact with the drug they were already taking. So you'd need to check for interactions with other drugs. There are many online reports making claims about the effects of chemical A. Some of these reports are biased, so based on opinion. Suggest one reason why a report making claims about the effects of chemical A may be biased. Now, biased, it has to be like someone is influencing those results. Why would someone want to influence the results and the report making claims about chemical A? So things like for financial gain. So whoever is making this drug could stand to make money, right, from selling the drug. So they might be making claims that it works more successfully than it does, for example, for financial gain. Or so competitors, maybe there are rival companies making alternative drugs, competitors suffer financial loss. Okay, because they might want to sell more of their drug and outcompete the competitors, and the competitors then have financial loss. But it has to be to do with finance here, because this is a drug that is going to be sold, and these individuals are biased. So they're going to be biased if they want to make money, right? Or they want to take money away from other competing companies. So it has to be something to do with finance, money, to get this mark, okay? How can scientists be sure that the claims about new drugs are valid? Now, valid means like accurate, correct, true. The drug has been tested thoroughly. All the variables have been controlled. So the results are valid. Advertising the claims on social media, well, that's definitely not proving it's valid. Ask an international company to produce the drug. Well, they might not test it thoroughly. They might also be biased. Have the claims peer reviewed, this one, because this means a peer or multiple peers, so other scientists in the same field, are going to check. They're going to test the drug and they're going to compare their results to your results and check that your claims are correct. That would be the best way to do it. Obviously, if you publish the claims in a newspaper, still might not be valid, still might be biased, but having the claims peer reviewed is the best answer here. Okay, this question is about the circulatory system. So figure 1 shows the human heart. Which part of the heart receives oxygenated blood from the lungs? Well, let's go through the options. In the right side of the heart, we've got deoxygenated blood coming into the right atrium and then deoxygenated blood coming out of the right ventricle going to the lungs. But over on the left side of the heart, we've got oxygenated blood coming in here through the pulmonary vein into the left atrium. So it's going to be A, isn't it? Because we're looking for which part of the heart receives oxygenated blood from the lungs. So it's coming in here as oxygenated blood from the lungs via the pulmonary vein into the left atrium. Which part of the heart pumps deoxygenated blood to the lungs? Well, that's going to be C, because we know the right side of the heart deals with deoxygenated blood, and that C is the right ventricle, which is going to contract and pump deoxygenated blood up to the lungs via the pulmonary artery. A group of cells found in the heart called the pacemaker controls resting heart rate. Where in the heart is the pacemaker found? Well, it's about here. So it's in the wall of the right atrium. So the correct answer here is right atrium. I have actually seen them ask students to put a little cross on the diagram as well for that question before. So anywhere in the right atrium would have been correct, because the pacemaker is actually found in the muscular walls of the right atrium. Figure 2 shows a cross-section of an artery and a vein. Describe 2 ways the structure of an artery is different from the structure of the vein. They've been quite kind because they've given us a diagram. They haven't said which one is the artery and which one is the vein, but we should recognise that this one is the artery and this one is the vein. And if we can, that's going to help us to answer the question. So, artery has thicker muscular walls or thicker muscular tissues. Artery has thicker elastic tissues or elastic walls or elastic fibers. And you can see those layers here. So you've got the muscle tissue here, you've got the elastic fibers here. Both of those layers are thicker. Now, if you don't get both of those marks, you can get 1 mark for just saying the artery has a thicker wall. But that would just be 1 mark, okay? You can also say the artery has a narrower lumen, which is the space in the middle of the vessel through which the blood flows. And you can see the artery's lumen is narrower compared to the vein's lumen, which is much wider. What else can we say? We can also say arteries do not contain valves, whereas veins do. Veins do contain valves. You do have to make this comparative. So notice that I'm using comparative language. For example, the artery has thicker muscular tissue. The artery has thicker elastic tissue. The artery has a narrower lumen. So I'm making sure that I'm clearly comparing it to the vein. If I don't say thicker, if I just say the artery has a thick, I'm not going to get the mark. I'd have to say the artery has a thick muscular wall, whereas the vein has a thin muscular wall. Or it's more easy to just say the artery has a thicker muscular wall. But I have to make sure I'm comparing my artery to my vein. Now, I could have done this question the total opposite way round. So I could have said the vein has a thinner muscular wall, or the vein has thinner elastic tissue. The vein can have valves, whereas the artery doesn't, or the vein has a much wider lumen. Obviously, the artery has a narrower one. Okay, right. Let's see what else we've got. In coronary heart disease, the coronary arteries become narrow. A buildup of fatty material can cause a blockage in the coronary artery. Table 2 shows how a blockage in the coronary artery affects blood flow. All we need to do is describe the trend. So when we're describing the trend, we should try to use the headings, the actual wording that they've given us in the table. So I'm literally going to do that. I'm going to use the headings. As the percentage of coronary artery that is blocked increases, because I can see that's increasing here, the blood flow decreases, which I can see here. okay? You could have said the opposite. You could have said as the percentage of the coronary artery that is blocked decreases, the blood flow increases. The greater the blockage, the less the blood flows, or you could say it's a negative correlation. But I always try to describe the correlation by actually using the headings in the table. But that's actually quite an easy question, isn't it, for 1 mark? Complete figure 3. So you've got a graph. You should use a suitable scale for the y-axis, plot the data from table 2, and draw a line of best fit. So they've already labelled the x-axis. They've already put the scale on the x-axis. They've labelled the y-axis, but they've not given us a scale. So we need to think of a suitable scale, and it has to go from 2 all the way up to 100. So let's think. I think if we go up in tens, yeah, that's quite a suitable scale. The reason that's a suitable scale is I'm using equal divisions. It's going to be really easy to plot because I've made it so 5 squares is equal to 10. So that's quite an easy scale. And in terms of the space, I have used at least 50% of the axis. So you do have to use at least 50% of the space that they've given to you, basically. Now you're going to get 1 mark if you give a scale like this on the y-axis. Then we can actually go ahead and plot the data. It's going to be a line of best fit. It is going to be a line graph as opposed to a bar chart because we've got continuous variables or numerical data on both axes. So I'm going to start with the first one. I'm plotting 0 and I'm plotting 100. So I'm going to do a little small x there. I'm plotting 10 and I'm plotting 64. I'm plotting 20 and 42. I'm plotting 50, make sure I'm getting the right place on the x-axis, 50, and I'm plotting 8. And then I'm finding 80 and I'm plotting 2. Okay, now if I plot all of my points correctly, I can get 2 marks. Okay, if I only plot 3 or 4 correctly, I'll just get 1 mark. Okay, so there are 5 plots in total. All 5 plotted correctly will get me another 2 marks. And then I'm going to get marking point 4 for drawing my line of best fit. Now, if I look at the pattern or the trend, it's clearly a curve. This is going to be quite hard for me to do on my drawing pad, but I'm going to try my best. Whoop, not the best. Let me try and do that last little bit again. It's really hard to get. You kind of need to get your arm inside the curve to do it, which I can't really do on my writing pad, but I've tried my best. Now, what we don't want to see is point to point. We don't want you to get your ruler and join them dot to dot because it clearly says a line of best fit. So it doesn't have to be perfect. It has to go close to all the points. So it should go within kind of a square, I would say, within a square. So close, very close to each plot. But it doesn't have to be perfectly through the center of all your plots. It should not be dot to dot, okay? So that's it. Then I can get 4 marks. Now, we shouldn't really extrapolate. Extrapolate means to kind of extend your line beyond the data that you have. We shouldn't really do that, although you're not going to get penalised for it here. But really, we should stop at 0 and stop at 80 because that's all the data that we've got. It should be a nice smooth line of best fit, and it is clearly a curve, so a nice smooth curve of best fit. Check your points are accurate because it's worth 2 marks for accurately plotting those points. Make sure you put the scale on your y-axis, make sure it's sensible, easy to plot, and make sure it fills at least 50% of the graph paper. And that's a really easy 4 marks. Predict the blood flow in a coronary artery with 35% blockage. I'm going to have to go back. So 35% blockage is here. So I'm going to predict based on my graph, it's about 23. I would say, let me just check that again. Yeah, based on my graph. So I'd get my ruler out in the exam. I'd draw a line up and then I draw a line across. So I've got some nice construction lines and I'm going to say it's about 23. centimeters cubed per minute. Now they will check your graph and they will check that you have given the correct answer based on your graph. And again, they'll give you tolerance of about plus or minus half a small square, just like they do with your plots on the graph. But yeah, for me, it's definitely 23. Explain the effect of a partly blocked coronary artery on the human body. This is a 6-mark question. So this is what we call an extended response question. So we're going to have to obviously simply give the effect, but we're going to have to make sure we lead it all the way through to a full explanation of how that will affect the human body to get ourselves up into 5 or 6 marks. So remember, the coronary artery is the artery that provides the heart muscle itself with blood. Just to remind you of that. Okay, it's the one that actually carries blood with oxygen and glucose to the heart muscle itself. So if we remember that, we can start right at the beginning of the story and think how, if that is partly blocked, that will affect the heart and then the rest of the human body. But we're going to start with how it's going to affect the heart because that's where this artery is. So if we partially or partly block the coronary artery, we're going to see reduced blood flow to the heart or the heart muscle itself. So less oxygen to the heart or the heart muscle or the heart tissue. So less glucose to the heart or the heart muscle or the heart tissue. So I'm just starting right at the beginning. It's the coronary artery. So if that is partly blocked, there's less blood getting to the heart itself. Blood obviously carries oxygen and glucose, so less oxygen, less glucose getting to the heart itself. So I've covered kind of the heart itself. So what does this mean for the heart? Well, it means less respiration of the heart muscle cells. You could say aerobic respiration, because obviously if it's getting less oxygen, it's going to be doing less aerobic respiration. Or at this point, you could just say less respiration of the heart muscle cells. Potentially, the heart cells do more anaerobic respiration. Because they are getting less oxygen, if they're doing less aerobic respiration, then potentially they're doing more anaerobic respiration. Anaerobic respiration releases less energy. So you could get that marking point down. If they're doing less aerobic respiration, obviously they're getting less energy. And if they're doing anaerobic respiration itself, then obviously they're getting less energy because anaerobic respiration is less efficient. It does release less energy. So less muscle contraction, because muscle contraction requires a lot of energy. And if the heart muscles themselves are doing less respiration or they're doing anaerobic respiration, they're going to have less energy released, so less muscle contraction. Now we can get to the rest of the body, and we're going to have to get this far to push us up into 5 or 6 marks. So if there's less muscle contraction of the heart, now we can get all the way to saying less blood or oxygen or glucose is pumped around the body because the heart muscle contractions are weaker or fewer. So we're actually going to see less blood or less oxygen or less glucose going around the body, which could mean less aerobic respiration. Now we're talking of the body, right? Of body cells, or more anaerobic respiration. Now we're talking about the body cells, remember? And then we need to get all the way to what that might cause. Well, it could cause tiredness, because if we're doing less aerobic respiration and more anaerobic respiration, again, we're releasing less energy. It could cause breathlessness, because we're going to try to breathe quickly or breathe more deeply to make up for that lack of oxygen that's getting to our body cells. It might, if we're doing anaerobic, lead to a buildup of lactic acid, which can lead to muscle fatigue, or muscle pain or muscle cramps. It may also lead to less CO2 removed from the body, because if our heart is not contracting as frequently or as strongly, less blood is flowing around the body. So the cells are not going to be able to get rid of their carbon dioxide as quickly. It's not going to get carried to the lungs as quickly. We're not going to breathe it out. This is a hard question. Let's just check we're happy with what we're saying here. We've got a partly blocked coronary artery. That's the artery that supplies the heart. So we're starting with the effect on the heart. If the artery that supplies the heart is partly blocked, less blood will flow to the heart. That's obvious. But that means less oxygen and less glucose is getting to the heart. That means the heart cells themselves are going to do less aerobic respiration and potentially might do anaerobic respiration. This means the heart cells themselves release less energy, and this means there's less energy for the heart muscle to contract. Now, if the heart can't contract as forcefully, then, and this is how we get up to 5 or 6 marks, we're going to have less blood flowing around the body. So less oxygen and glucose getting to body cells. So now body cells can't do as much aerobic respiration or have to do anaerobic respiration. Anaerobic respiration, we know, leads to a buildup of lactic acid. It leads to muscle fatigue or muscle cramp. We might also be lacking in energy, so we feel tired. We might be breathless because we're trying to get in more oxygen, and we're going to breathe out less CO2 because the blood is not getting it to the lungs as quickly. Now, for level 3, students must explain the effect of reduced oxygen or glucose on respiration. They've got to mention respiration. They've also got to convey the idea that less energy will be released, and they've got to give some kind of consequence for the body. So things like this: tiredness, breathlessness, buildup of lactic acid, muscle fatigue, or less oxygen removed from the body. I would kind of split my answer into 3, like the effect on the heart, the effect on blood flow around the body, and then a consequence on our body, like how we actually feel or what would actually happen. But yeah, not an easy question, so I hope that explanation has been useful. Let's look at 2.9. There are different treatments for blockage in a coronary artery. Explain how one treatment for a blockage would work. I think you're probably going to go for use of stents. And if you go for use of stents, you'd have to say to open up the coronary artery, or keep the coronary artery open or wide in the coronary artery, because the stent is inserted into the artery. It's a mesh-like tube that pushes out against the walls, holds the coronary artery open so more blood can flow. So you're probably going to go for that one. But you could also go for prescribe statins, because they help to reduce blood cholesterol. So hopefully that will over time lead to the blockage getting smaller and also no more plaque being added to that blockage. What else could you go for? Heart transplant, would you go for that one? You could do, it's unlikely you're going to go for that one. But heart transplant, obviously with the coronary artery still attached, to replace the diseased heart with a healthy heart. Probably not going to go for that one. You're probably going to go for this one, I should think. You could even say carry out a heart bypass where other blood vessels are connected so the blood can bypass the blocked vessel. So you can take a blood vessel from somewhere else in the body and connect that, and then the blood doesn't have to go through the coronary artery. It can go through the new vessel or the bypass. But you're probably going to go for this, and this is an easy 2 marks. Just name the treatment and then describe briefly how that treatment works. Question 3, Salmonella bacteria causes outbreaks of food poisoning in humans. To prevent food poisoning in humans, farmers vaccinate their animals against Salmonella bacteria. How do Salmonella bacteria in food cause the symptoms? Now, you might be questioning what they want here, but this is a bacteria. And at GCSE, we learn that bacteria produce toxins. That's what causes the symptoms. You can say produce toxins, you can say release toxins, you can say poisons instead of toxins, but that's how bacteria make us ill. So that's all they want. During a food poisoning outbreak, scientists identified the farm where the food came from. The farmer had not vaccinated the animals against Salmonella bacteria. The food poisoning outbreak could have been prevented if the farm animals had been vaccinated. Explain how. The immune system of animals respond to vaccination, and the immune system response in farm animals prevents an outbreak of food poisoning in humans. 4 marks. I think this is a really hard question. We need to kind of split it into 2. We need to think about how initially the animals respond to vaccination and how that response in animals will then help to prevent an outbreak of food poisoning in humans. So this is a big, tough immune response question. Okay, so if we vaccinate an animal, let's start there. If we vaccinate an animal, so we're going to do this bit first, what happens in the animal? So the animal's white blood cells, or you might know the word lymphocytes, but you can just say white blood cells for GCSE. They will produce antibodies against, you can say Salmonella, you can say against the vaccine, you could even say against the antigen, because you know it's the antigen on the surface of the pathogen that is going to cause that immune response. So white blood cells produce antibodies against the Salmonella. Okay. What's going to happen next? Once they've produced the antibodies, I think it's worth us saying that the antibodies are specific or complementary to the Salmonella or the Salmonella antigens, okay? So they bind to the Salmonella or the Salmonella antigens, okay? So it's not just any old antibody, it has to be the specific or correct antibody that is complementary to those specific antigens on the Salmonella that would have been in the vaccination, a weakened or dead or inactive form. Okay, so what's going to happen next? If infected again, and this is where the immunity from the vaccination comes into it, if infected again, antibodies are produced more quickly or in larger numbers or in higher concentration. So if the farm animal is infected for real with Salmonella, that farm animal, because it's had the vaccination, is going to be able to produce the antibodies far more quickly or in larger numbers. And remember, this is the white blood cells that are producing antibodies. I haven't repeated it here because I've already said right at the start that it's white blood cells that produce antibodies, okay? But they're going to be produced more quickly or in larger numbers. So the antibodies destroy or kill the Salmonella, okay? Which ultimately means there'll be fewer Salmonella bacteria in the animals. Because if they ever get infected, the white blood cells are going to be able to produce these antibodies super quickly and in large numbers and destroy the Salmonella. So the numbers of Salmonella will be much, much lower. So there's going to be far fewer Salmonella in the animals or in the animals' products. So in the animal meat, in the animal milk, in the animal eggs. okay? So now we're going to be able to link it to humans, aren't we? Food poisoning in humans. Because if the farm animals are immune, if their white blood cells are quickly producing antibodies to destroy the salmonella, there's fewer salmonella in the animal, in the animal's meat, in the animal's milk. What that means is fewer salmonella bacteria in the animals when humans eat them. Or you could say when humans eat their meat, or when humans drink their milk, or when humans eat their eggs. There's going to be fewer salmonella bacteria in the animals when humans eat them. What else could we say? So, not enough bacteria consumed to cause infections in humans. Something like that. Not enough bacteria consumed to cause infections in humans, or not enough bacteria to produce enough toxins to make the human actually feel ill. Now, this is an extremely high demand question. You might think that I've written a lot, but to get 3 or 4 marks, you had to get all the way to preventing an outbreak in humans. So you had to say at least one of these points that I've written up here. You couldn't just talk about the response in animals. You couldn't just talk about what the vaccination does in animals or the secondary response in animals if they get infected again. You had to get all the way to why is this then likely to cause food poisoning in humans? So you had to convey the idea that, well, if there's less salmonella in the animals, there's going to be fewer bacteria in the foods that the humans consume when they eat animal products. And if there's less bacteria in the foods that humans consume, there's going to be less bacteria in their bodies to cause infection or producing toxins. So they are also less likely to get ill. Very few students got 3 or 4 marks on this question. Most students got 1 because they just talked about, well, the farm animals, you know, the white blood cells produce antibodies. The antibodies destroy the salmonella. That's just going to get you 1 mark, okay? If you went on to say, oh, and then they're immune, so if they get infected again, the antibodies are produced quickly to destroy the salmonella, you might get 2 marks. But to push yourself up into 3 or 4, you've got to mention something about the humans eating the food and being less likely to develop food poisoning. Very, very, very difficult question. Okay, most cases of food poisoning do not need to be treated with antibiotics. However, some patients may need to take antibiotics to recover. Scientists investigated the effectiveness of 5 different antibiotics in Salmonella bacteria in the outbreak. Antibiotics A, B, C, D, and E were used in the investigation, and figure 4 shows the results. Describe 2 aseptic techniques, so these are like our sterile techniques, that the scientists should have used in the investigation. There's absolutely loads. Things like disinfect hands or disinfect work surfaces. We're obviously using a Petri dish, so sterilize the Petri dish or sterilize the culture medium or the agar, which is what you're growing the bacteria on in the Petri dish. Pass inoculating loop through a flame. The inoculating loop is actually what you spread the bacteria with. It's made of metal, so we put it into a really, really hot flame to destroy any other bacteria, like contaminating bacteria that might be on that inoculating loop. Work in a fume cupboard, because then any bacteria that might be in the air are going to be moved up and sucked up into the fume cupboard. You can even say work next to a lit Bunsen burner, because as the hot air rises, it's going to carry up the bacteria that might be in the air. So the bacteria would get carried upwards in that current rather than falling down onto the Petri dish and contaminating the Petri dish. Pass forceps through a flame. Again, forceps are made out of metal, so the way that we sterilize them is to pass them through a hot flame. Tilt lid of Petri dish and open for a short period of time. This is to prevent, again, any bacteria from the air potentially falling on the agar and contaminating the plate. So you open it for as brief time as possible and you hold the lid at an angle so the bacteria can't get in. Secure lid with sticky tape or adhesive tape or sellotape. Obviously, so no one can open it easily, the lid can't fall off, and then potentially all the bacteria can get into the Petri dish and bacteria can get out and make you ill as well. Look, there's absolutely loads of things you can say. You could even, if you're using like a glass spreader, you can put your glass spreader in ethanol because that would sterilize the glass spreader. If you're using anything metal, we tend to pass it through a flame to sterilize it that way. Disinfecting hands, disinfecting working surfaces, securing the lid with sticky tape. These are all standard aseptic techniques. What we can't just say is clean the surfaces, wash the surfaces, wash our hands. That's not specific enough, okay? Sterilizing or disinfecting the surfaces, sterilizing or disinfecting our hands would be okay, but just washing or cleaning is too vague, okay? Sterilize, disinfect. That's the words we need to be using. We needed 2 of these. The scientists incubated the bacteria at 37 degrees Celsius. Students in school labs incubate bacteria at only 25. Explain why scientists use 37, but students must use 25. So there's kind of two points to this question. Why do scientists use 37? So 37 degrees C is human or body temperature. That's why scientists use it, because salmonella or the bacteria grow fastest or best at 37 degrees C. Remember, this is a bacteria that infects humans. It grows really quickly at 37 degrees C because that's human body temperature. So scientists are going to want to use 37 degrees C because that's human body temperature. It means the bacteria will grow the fastest, so they'll be able to study them faster. So we've got 2 marks for that, but why do students only use 25? So 25 degrees C is a lot cooler, right? So it prevents the growth or restricts the growth of bacteria that may be harmful to humans, such as salmonella. So we don't want students in a school to be encouraging the growth of human pathogens. So we don't want to use 37 because that would encourage the growth of potentially dangerous pathogens that are dangerous to humans. So we use 25, which is cooler. It's far below human body temperature. So we don't grow any bacteria that could potentially be harmful to humans. You could also say it's too low or too cold for the growth of human pathogens. Okay, so hopefully we're not growing anything dangerous. What is the purpose of the paper disc with no antibiotic in figure 4? This is what we call a control. It's not a controlled variable. Do not put control variable. It's a control or a control experiment, which means it's used to compare to the other discs to show the effects of the antibiotics. So one of the paper discs has got no antibiotics. So we can compare that disc and what happens to the bacteria around that disc to the other discs that do have antibiotics. And then it's kind of like we are truly able to see the effect of the antibiotic. We're truly able to prove it is the antibiotic that's killed the bacteria and not just the paper disc itself. So I mean, it's only 1 mark. So literally all you had to say was it's a control or it's a control experiment. That's all you had to say. But if you forgot that term, you could say something like it's used for comparison, or it's used to show that the results are due to the antibiotic and not just the paper disc. But we cannot say control variable. It's not something that we keep the same to make sure it doesn't affect the results. It's an experiment that we use to compare with the real experiments. And in this case, we're trying to prove it is the antibiotic that's killing those bacteria and not something else. Like, it's not just something else in the paper. It is the antibiotic that's killing those bacteria. The scientists concluded that either antibiotic A, which was this one here, or antibiotic B should be prescribed to patients with food poisoning. Why should antibiotic A or B be prescribed? Easiest way to get the mark, they have the largest zone of inhibition. This is what we call the zone of inhibition. Zone of inhibition. The zone of inhibition is the zone or the area where the bacteria has been killed, in this case by the antibiotic. So we could easily get 1 mark just for saying they have the largest zone of inhibition. If we don't know that phrase, we can say they killed the most bacteria. Yeah. We can't just say clear zone, by the way. If you've heard clear zone, that's not good enough. We say largest zone of inhibition, or they have killed the most bacteria. Because look, you can see all of this area, all of the zone of inhibition is where the bacteria has been killed, and the area is much bigger for A and B than it is for, say, E or D. The scientists wanted to be more certain about which antibiotic should be prescribed. Describe how the results in figure 4 could be used to obtain a quantitative comparison. Quantitative means numerical data. So what we're going to have to do here is actually measure the area. It looks like the zone of inhibition is bigger for B than A, but what we could do is actually measure that circular area to get quantitative data. Now, how do we find the area of a circle? Well, you actually don't need to go that far. You could simply just say, measure the diameters, and the one with the biggest diameter has the biggest zone of inhibition. You could say measure the radius of the circle, and the one with the biggest radius obviously has the biggest zone of inhibition. Or you could go as far, which is what I was going to say, is calculate the area of each clear circle, or the area of each zone of inhibition. Now, obviously, to calculate the area, you'd have to know the radius, because you do pi times the radius squared to get the area of the circle. But all it says is we want a quantitative or a numerical comparison. So we could simply just say, well, measure the diameters and compare them, measure the radius and compare them, calculate the area and compare them, and measure the diameter or the radius where the bacteria have been killed, measure the diameter or the radius of the zone of inhibition of the clear area. But yeah, you might have actually learned in school that once you've got the radius, you can do pi r squared to get the whole area of that zone of inhibition. Obviously, the one with the biggest area, the biggest zone of inhibition, is going to be the best one to prescribe because it's killed the most bacteria. 1 year later, there was another outbreak at the farm involving Salmonella bacteria. Antibiotic B did not have an effect. Suggest why antibiotic B no longer had an effect. Yeah, it looks here like antibiotic B was super effective at killing the bacteria, but now it's saying it's no longer effective. So antibiotic B, let's just word this differently. The bacteria, must be resistant to antibiotic B. All they're really testing you on here is that you use the word resistant. Some students keep falling into the trap of saying the bacteria must be immune. They don't have an immune system. They don't use white blood cells. They can't get immune. They're not becoming immune. They are developing resistance. So the bacteria are becoming resistant or have become resistant to the antibiotic, which means they can no longer be killed by the antibiotic. Antibiotics treat food poisoning because they kill Salmonella bacteria inside the human body. Some antibiotics work because they damage the bacterial cell wall, and the bacteria die because the cells burst. Ooh, this is a good question. This is actually about osmosis, but it's a little bit sneaky. The reason I know it's about osmosis is because we've got cells bursting and we've damaged the bacterial cell wall with the antibiotic, and the bacteria die because they burst. So why would the cells burst? So it's because water enters the bacterial cells by osmosis for marking point 2. Then why do they die? Well, the cell wall is damaged, okay? So the damaged cell wall cannot withstand the pressure. So they burst. And that's it, really. So water's going in by osmosis, because osmosis is the movement of water from a dilute to a concentrated solution. So water's going into these bacterial cells by osmosis. Just saying that word can get you another mark. And then why does this destroy the cells or cause them to burst? Well, if they're filling up with water, the pressure of that water pushing against the membrane, pushing against the cell wall, if the cell wall is damaged, it cannot withstand that pressure. So the cell is going to burst. And that is 3 marking points. To question 4. So this question is about exercise. During vigorous exercise, we've got anaerobic respiration occurring in a person's body. Explain 2 effects of anaerobic respiration. So that's why it's 4 marks, because we're not just stating the effects, but we're explaining the effects. So we need to give an effect and then explain it. So for example, an effect might be that the person, if they're using anaerobic respiration, could get muscle fatigue. And the explanation for that would be because it's caused by a buildup of lactic acid. So this is the effect on the body. This is the explanation of the effect. Instead of muscle fatigue, you could have said the effect would be oxygen debt occurs, but that would have the same explanation. The reason we get an oxygen debt is because there's a buildup of lactic acid, which we need to break down by repaying the oxygen debt or using oxygen to break down the lactic acid. Another effect on the person's body, even after exercise, you'd see increased breathing rate, or you could say heavier breathing, deeper breathing. That would be the effect on the body. The explanation for that would be to repay the oxygen debt, or you could say to provide oxygen needed to break down the lactic acid. Because that's what the oxygen debt is. You have to repay the oxygen debt. You need oxygen to break down that lactic acid. Similarly to that one, increased breathing rate, you could also go with increased heart rate. Even after exercise, heart rate stays high. So continued increased heart rate or just increased heart rate. But again, you would link that to the same explanation. So it has to stay high to repay the oxygen debt or to provide the oxygen needed for the breakdown of lactic acid, because obviously oxygen is carried in the blood. So by keeping our heart rate up, it's going to deliver plenty of oxygen to the muscles so that oxygen can be used to break down the lactic acid. So that would be the effect, but you'd use the same explanation for that one. 4th option, an effect could be that we see fewer or weaker muscle contractions. That would be the effect on the body. So the explanation would be because anaerobic respiration releases less energy. Obviously, it's less efficient than aerobic. It releases less energy than aerobic because the glucose doesn't fully get broken down. So because there's less energy available, you'd see fewer or weaker muscle contractions. Now, you didn't need all 4 of these. You needed 2 effects with 2 reasons, but they will be marked in pairs. So you can't mix and match. You have to match the correct reason to the right effect. Okay, part 2. design an investigation to show the effects of different types of exercise on the heart rate of athletes. The first thing I do with these kind of questions is try to identify the independent variable, so the one that I'm going to need to change to see the effect of it, and then try to identify the dependent variable, the one I'm going to measure and record, and then obviously I'm also going to think about controlled variables. So if I got this question, the first thing I would do is highlight this. The independent variable is going to be the type of exercise. So I'm going to need to use different types of exercise, different intensities of exercise. I could do jogging versus running, for example, or running versus cycling, or swimming versus running. And what am I going to be measuring? Well, I'm going to be measuring the heart rate of athletes, but I do want to show the effect of the different types of exercise. So I'm going to have to think about measuring the heart rate before and after exercise, so I can actually see if there's an effect, if there's a change in the heart rate. So now I've done that, I think I can start the question. So I'm going to have to get a large group of athletes. Remember, with any investigation, we don't just want to do one repeat, we want to do many repeats or have a large sample size. So I'm going to need a lot of athletes. If you're going to give a number, I would go for 30 or more. That's what the mark scheme said for this question. That's what they're classing as a large number. So 30 or more athletes, large group of athletes. Get them to do different exercises. Now, you don't have to specify which exercises, but you could say, get them to sprint, get them to jog, get them to swim, get them to cycle. But you need at least 2 different exercises if you are going to give examples, because you have to make a comparison between the effect of different types of exercise. That is our independent variable. So you are going to have to get these athletes to do at least 2 different types of exercise. You're also going to have to measure their heart rate before exercise and after exercise, because you want to see the effect on their heart rate. So if you don't know what it was at rest, how are you going to know the effect? So you're going to have to record their heart rate before and after exercise. Now, if you don't get this key information down, you're going to be stuck at 2 marks, because you're not going to see the effects of exercise unless you've done the heart rate before and after. Okay, then you can calculate the increase in heart rate. And you could also calculate the mean increase in heart rate for your group of athletes. Say you've got 30 athletes, you could record each individual's increase in heart rate and then work out the mean increase in heart rate. Or you could, if you're not calculating the increase in heart rate, you could say things like compare heart rate before and after exercise. So we're just trying to make it clear. You're either going to have to work out the difference between the 2 heart rates before and after, or at least say compare their heart rates before and after exercise. But ideally what you do is you'd measure every single athlete's heart rate before, every single athlete's heart rate after exercise. Then you'd work out the difference in each athlete's heart rate, and then you could find the mean difference in heart rate to see if it's gone up on average or stayed the same or gone down maybe, depending on the intensity of the exercise. Now, if we want to make sure we get 5 or 6 marks, we need some control variables. I always say give at least 3. So think about things that might affect the results if you did not control them. So all your athletes, you could say they should be the same sex, they should be the same age, they should have the same kind of health issues, or not the same, maybe say no health issues or no illness, because if you've got some athletes that, I don't know, have a heart condition or have asthma, that could affect the results. So ideally you want them all to be of the same level of health, the same level of fitness, or have no health issues, no illness. What else could we say? Same length of time for each exercise, because if they do one exercise longer than the other exercise, that would obviously make their heart rate increase more. So that would then affect your results. You could also say, make sure their heart rate returns to normal between each exercise. So say, for example, if you're getting them to jog and then you're getting them to sprint, you'd have to get them to jog, record their heart rate before and after, wait for it to get back down to normal before you then get those athletes to sprint. Because obviously, if their heart rate hasn't gone back down to normal, it's going to already be elevated and that's going to affect the results. Remember, control variables are all about controlling things, keeping things the same that could ultimately affect the results. The results here are the effect of heart rate, the change in heart rate. All of these things I'm listing here could potentially affect heart rate if they are not controlled. Anything else? Same caffeine intake even was on the mark scheme. Make sure they're on no medication, because that, if some of the athletes are taking certain drugs or certain medications, that could affect their heart rate. So yeah, that's how we're going to get 6 marks. If we make it clear to the examiner, look, we're going to use a lot of people, a lot of athletes. We are going to change the exercise. We're going to do different exercises, or at least 2 different exercises. We're going to measure their heart rate before. Then they're going to do the exercise. We're going to measure their heart rate after. We're going to calculate the change in heart rate. We can then calculate the mean change in heart rate or compare the heart rates before and after. And we're going to give at least 3 control variables. So things that if we didn't keep the same would affect the heart rate. You can see I haven't really written a method. I've kind of just thought, well, what's my independent variable going to be? How am I going to change that? What's my dependent variable going to be? How am I going to measure that? What are my control variables going to be? Give at least 3, and then make sure that I've got plenty of athletes, so I'm doing repeats. So I always teach my students, think about the IV, the DV, the CV, and repeats, okay? What are you going to change and how? What are you going to measure and how? What are you going to control and why? Think about repeats or a large sample size in this case, and then we'll get 6 marks. Okay, anabolic steroids are drugs. Anabolic steroids increase muscle mass in humans and are banned in most competitive sports. Some athletes take anabolic steroids to improve their performance. Explain how taking anabolic steroids could improve an athlete's performance. Well, it's kind of giving us a clue here, right? They're going to increase muscle mass. We can't just repeat that because it told us that in the question, but we can use that to think about how it might improve their performance. So things like athletes would be stronger because they've got increased muscle mass, potentially faster, like if they're running, the muscles in their legs might be stronger, they'll be able to run faster. You could say things like they might have increased stamina, they might have increased endurance, or any other kind of description of improved performance. So they might be able to lift heavier weights. I don't know, any other description of improved performance. But what we can't do is just repeat what was in the question. We have to think about how that might actually improve their performance. And the second mark, increased or stronger or more muscular contractions. Okay, because we know they've got increased muscle mass, so they're going to be able to do stronger or more muscle contractions, and this is ultimately what's going to make them stronger or faster or increase their endurance or their stamina. So they were the 2 marking points for that one. Scientists use monoclonal antibodies to test for the presence of anabolic steroids in an athlete's urine. Now, monoclonal antibodies, remember, are produced by cloned lymphocytes. So they'll all be exactly the same shape and they'll all bind to the same antigen or the same protein, or in this case, they'll all bind to the anabolic steroid. To produce monoclonal antibodies, a mouse lymphocyte is combined with a tumour cell. What type of cell is created when the lymphocyte binds with a tumour cell? It's called a hybridoma. So that's just a multiple choice question. Describe how the scientists make monoclonal antibodies using the cell created when the mouse lymphocyte and the tumour cell combined. So using the hybridoma. So we're not going to get marks for saying take a mouse lymphocyte and fuse it with a tumour cell to create a hybridoma, because we've already got that in the question. So once we've got the hybridoma, what we then need to do is finish the story and talk about how we actually get the monoclonal antibodies. So we can say things like hybridoma is cloned, which means many identical cells or identical hybridomas will be produced, and all these cells make the same antibody, or the specific antibody that in this case will be complementary to the anabolic steroid, because they all came from the same hybridomas. Yeah, we took one specific lymphocyte which was creating the right antibody. We fused it with a tumour cell that divides rapidly. So we've now got a hybridoma that has both of those properties. It can divide rapidly, it can clone itself rapidly, and it will have the characteristics of the lymphocyte, so it will be able to produce the specific antibody that we need. The hybridoma will clone itself, will have many identical cells. All of those cells will now make the correct or the specific or the same antibody. And finally, we can say the antibodies are then purified, or we often say collected and purified, but the mark is for saying purified, because then we've got to that final part of actually how do we get the monoclonal antibodies. They'll all be the exact same type of antibody because they were all produced from the same original lymphocyte that was making that specific antibody. Okay, so that's how we get 3 marks. There's 4 bullet points there, but you needed 3 of them to get 3 marks. What property makes a monoclonal antibody useful in detecting the presence of an anabolic steroid? So the monoclonal antibody is quick and easy to produce. Well, not really. We've just seen it's quite a complex process using the lymphocyte and the tumour cell to make the hybridoma, which then has to clone itself. We have to collect the antibodies and purify them. A monoclonal antibody is specific to only one person's urine. Well, no, we could use it to detect the presence of this anabolic steroid in anybody's urine. A monoclonal antibody only binds to the anabolic steroid. That sounds right to me. And it definitely can't identify many different drugs. Remember, they're all the same type of antibody. They were all produced by the same lymphocyte. So they're all going to be complementary to just one specific protein or one specific antigen, or in this case, just to one anabolic steroid. This is why they're so useful at detecting that anabolic steroid, because they only bind to that anabolic steroid. So the answer there is the third statement. Figure 5 shows a test strip that can detect the presence of the anabolic steroid in the athlete's urine. The end of the test strip, so here, is dipped in the urine. The urine will move up through the test strip. The test area and the control area both contain a dye, and the dye turns blue when monoclonal antibodies bind to it. Suggest the purpose of the control area. Now, the control area is the same as with a pregnancy test. It's there to show or to prove the test is working. Because if there are anabolic steroids in the person's urine, or there's not anabolic steroids in the person's urine, it shouldn't matter. You should always get the dye turning blue in the control area, because any free monoclonal antibodies, and we know the test strip contains free monoclonal antibodies, it says it here, they should move up the test strip as the urine diffuses up the strip. So there should definitely be free monoclonal antibodies binding here and turning the dye blue. So it's just to prove that the test is actually working. It's the same with the pregnancy test. That's always what the control area is for. Figure 6 shows the urine test results for 4 athletes. Describe the evidence in figure 6 that shows that the test for athlete B has not worked, and suggest one reason why the test did not work. Well, if you look at the control area, which was here, you can see there is no blue dye or no visible dye in the control area. So it obviously hasn't worked. You could also say there's no coloured line in the control area, there's no colour change in the control area, but we should always get blue dye in that coloured area. It's showing us that the test is working because there should always be free monoclonal antibodies that will bind to that control area. Suggest reason why the test did not work. So maybe no monoclonal antibodies bound to the control area. What else could we say? Maybe there were no free monoclonal antibodies at the end of the strip. Because if we go back to the previous diagram, it said that there were free monoclonal antibodies. Maybe if there weren't any, the test was faulty, there weren't any free monoclonal antibodies, then they wouldn't have reached the control area, and that's why the dye did not change colour. What else could we say? Maybe the free monoclonal antibodies did not move up. Maybe they were kind of fixed in place or locked in place, which was, again, a faulty test. Did not move up the test strip. And therefore, they can't bind to the control area and can't make the dye go blue. Maybe the urine did not move up the test strip. Maybe because there wasn't enough urine applied, so the urine didn't move up the test strip, so it didn't carry the monoclonal antibodies up the test strip, and that explains why there's no blue dye in the control area. Which athlete had a positive test? Well, it's going to be D, right? That's kind of obvious. The reason we know it's going to be D is, look, we've got the test area here, and we've got the control area here. Now, the control area is just to check it's working, so that should always turn blue if it's working. The test area contains a chemical that binds to monoclonal antibody steroid complex. So it will only turn blue if monoclonal antibodies have bound to the steroid. So it will only turn blue if there were steroids in that person's urine that bound to the monoclonal antibody that would then bind to the test area. So D, it was obviously working because the control area has gone blue, and there was obviously steroids in the person's urine because the test area has gone blue, so it's got to be D. Question 5. The protist that causes malaria is passed from one person to another by mosquitoes. What term describes an organism that passes a pathogen from one person to another? The biological term for that is a vector. So it's transferring that malarial protist from one organism to another. Figure 7 shows the malarial protist. The malarial protist is a eukaryotic cell. Describe 3 ways the structure of the malarial protist, which is eukaryotic, is different from the structure of a prokaryotic cell. Do not refer to size in your answer. So we can't talk about size, but we can talk about anything else that we can see on the diagram. So the protist, which is a eukaryotic cell, has a nucleus. Okay, obviously a prokaryotic cell would not have a nucleus. The protist has, I'm just looking at the picture, mitochondria. So I can see the nucleus here, and I can see the mitochondria here. Now, prokaryotic cells do not have a nucleus. That's what prokaryotic means, basically. No nucleus, no membrane-bound organelles. So the protist has a nucleus, prokaryotes wouldn't. The protist has mitochondria, prokaryotes wouldn't. You could say the protist does not have a single loop of DNA, whereas a prokaryotic cell, it doesn't have a nucleus, it has loops of DNA in the cytoplasm. You could say the protist does not have plasmids, because most prokaryotic cells have plasmids, which are smaller loops of DNA in the cytoplasm. Not all prokaryotes have them, but at GCSE we kind of assume that they do. Now here with the protist, we can't see any plasmids, they don't have plasmids, whereas prokaryotic cells do have plasmids. What else? Protist does not have a cell wall, whereas all prokaryotic cells have a cell wall. I think that's the main things, isn't it? So it has a nucleus, prokaryotes don't. It has mitochondria, prokaryotes don't. It doesn't have a circular loop of DNA, prokaryotes do. It doesn't have plasmids, prokaryotes do. It doesn't have a cell wall, prokaryotes do. You need to be a little bit careful saying, some students will say things like, the protist doesn't have flagella. Well, not all prokaryotes have flagella. So we can't say that. Not all prokaryotes have them, only some. Some students will talk about ribosomes, but we can't talk about ribosomes because protists have ribosomes and prokaryotes have ribosomes. So they both have ribosomes. Weirdly, they did allow this, which is a little bit strange. They allowed the protist does not have a slime capsule. It's a little bit weird that they allowed that because not all prokaryotes have a slime capsule. Some of them do, but not all. So it's a little bit weird that they allowed that one. I wouldn't have gone for that one because not all prokaryotes do have a slime capsule anyway. I would have gone for like really obvious ones like these ones because no prokaryotes have a nucleus, no prokaryotes have mitochondria. So that's a clear difference. And all prokaryotes have a cell wall, and this protist clearly doesn't. So they are the best differences, I would say, to go with. During one stage of malaria infection, the malarial protists enter red blood cells and cause them to burst. Explain why the bursting of red blood cells causes tiredness. Well, we should know that red blood cells carry oxygen, right? They actually contain hemoglobin, which carries oxygen. But the gist of this question is, they carry oxygen. So if red blood cells burst, what that means is less oxygen is carried in the blood. We can get 1 mark for that. We can say less oxygen will be carried to cells, or less oxygen will be carried in the blood. And then second marking point, why is this going to lead to tiredness? Less oxygen for respiration, because we know that cells need oxygen for aerobic respiration. So we've got a link to tiredness, less energy released. If there's less respiration, there's less energy released. That's going to make us feel tired. Be really careful not to say produced. Energy cannot be produced. Yeah, that's the fundamental law of energy conservation. Energy cannot be created or destroyed. So we should never, ever in biology GCSE be saying energy is made or energy is produced, okay? What we say is that respiration releases energy. So if less oxygen is being carried to the cells or less oxygen is being carried in the blood, then there's going to be less oxygen available for respiration, so less energy will be released, which explains why it causes tiredness. 5.4, the malarial protist reproduces sexually and asexually during its life cycle. Complete table 3 to give 3 differences between sexual and asexual. One difference has been done for us, so we can't use that one. Sexual involves 2 parents, asexual involves 1 parent. We can't use that one. But what we can say for sexual reproduction, we could say it involves gametes fusing, or as an alternative to that, we could say it involves fertilization, which is when the gametes fuse, whereas asexual, no fusion of gametes, or the same marking point would be no fertilization. So there's no sperm fusing with an egg because it's just one parent just using mitosis to cause its cells to divide to grow into a new organism. So that would be 1 marking point. With sexual reproduction, the offspring will be genetically different Or you could say sexual reproduction introduces genetic variation. But the word genetic or genetically is important. If you just say the offspring will be different, that's not good enough. If you just say it introduces variation, that's not good enough. It's genetic variation or genetically different. With asexual reproduction, offspring will be genetically identical, or you could say there is no genetic variation, because obviously the offspring are getting all of their DNA from one parent, right? So they'll have the exact same DNA as their parent, so they'll be clones. Marking point 3, sexual reproduction involves meiosis because it involves gametes, and gametes are made by meiosis, whereas asexual reproduction involves mitosis. The new organism is produced just via normal cell division, mitosis, which produces genetically identical cells. There are other answers we could give, like for sexual reproduction, more energy is required because it involves finding a mate, it involves fertilization. Sexual reproduction requires more energy. Asexual reproduction requires less energy. And the final one, sexual reproduction is slower, it takes more time. Asexual reproduction is faster. So there are 5 answers. You must make sure they're comparative. So your boxes must kind of line up with comparisons or differences in this case. Yeah, and you needed 3 of them to get 3 marks. One drug for testing malaria prevents mitosis occurring in the malarial protist. The drug stops the synthesis of new DNA bases in the cell. Suggest how the drug prevents mitosis occurring. Well, if we can't make DNA bases, we can't make any new DNA. So what's basically happening here is the drug is stopping DNA replication, or DNA replication cannot occur because we can't make any new nucleotides, we can't make any new bases, so we can't build any new DNA. So DNA replication cannot occur, or you can say DNA cannot be duplicated, or the amount of DNA cannot be doubled, something like that. And obviously if the DNA can't be replicated, then mitosis or nuclear division cannot occur. Describe the process of cell division by mitosis. This is a nice question. This is just one where you have to learn the story of mitosis. So we've already had DNA replication because we've mentioned it in the previous part of the question. That happens during interphase. Then we go into mitosis, which is nuclear division. So once we've replicated the DNA, what we should be saying is one set of chromosomes is pulled or is moved to each opposite end or pole of the cell. Now you have to be really specific with your language here. For example, you can't just imply that the chromosomes are moving on their own. You have to convey the idea they're being pulled, because it is the spindle fibers that are doing it, right? They're being pulled or moved. You also have to convey the idea that they're going to opposite ends or opposite poles of the cell. So one set of chromosomes will go that way, one set of chromosomes will go that way. They're being pulled to opposite ends or opposite poles of the cell. Then after that's happened, the nucleus divides, or you can say 2 new nuclei form, because remember, mitosis is nuclear division. So the nucleus divides, or as an alternative, 2 new nuclei form. That's all we can say for that point. Finally, the cytoplasm and cell membranes divide to form 2 genetically identical cells. Or if you know the key term, it's easy to just say cytokinesis occurs. So the cytoplasm divides, the cell membranes divide, or I think I actually would have said it like this. I would have said the cytoplasm divides and 2 new cell membranes form. I think that's how I would have said it. But yeah, the cytoplasm divides, the two new cell membranes form, or the cell membrane divides, and you will form two genetically identical cells. This is called cytokinesis. That's how we get cell division. Okay, really, really tricky question. You've got to be so specific with your wording for that one. Okay, different types of disease may interact. So scientists studied the incidence of malaria infections in children with disorder S and without disorder S. So we're looking at the number of cases of malaria in children with a different disease or without the different disease. The incidence of malaria in children with disorder S was calculated as a percentage of the incidence in children without disorder S. And table 4 shows the results. Describe what the results in table 4 show about the interaction between disorder S and malaria. Well, the first thing that I think is really easy, and remember this is just to describe, so we're really just describing the trends in the data. You can clearly see that, oh, actually it's not that straightforward, is it? It's not that straightforward. As I was going to say, as age increases, the percentage incidence of malaria increases, but it's not that straightforward. It's actually decreasing up to the age of 10, and then it's increasing, isn't it? Okay, so let's write that down. It's quite a hard question, this one. So as age increases, so going from like 2 to 10, as age increases, there is a decrease in the percentage of children with disorder S who have malaria. I'm just using the kind of words in the top of the table there. So as age increases up to the age of 10, by the looks of it, the calculated percentage incidence of malaria in children with disorder S is decreasing. So as age increases, there is a decrease in the percentage of children with disorder S who have malaria. That's fine. Up until age 10. Then the percentage increases. Does this make sense, guys? This is quite hard, isn't it? So we're looking at the age. As the age is increasing, the percentage incidence of malaria is decreasing in children with disorder S. But then once we get to age or above age 10, it goes up again. The percentage incidence of malaria goes up. So as the age increases, there is a decrease in the percentage of children with disorder S who have malaria up until age 10, then the percentage increases. Okay, I think we got there in the end. Let me just find my pen. Question 6, this is the last question. This is about photosynthesis. I love how they tell you which topic each question is about. I don't know whether you've noticed that as we've been doing the test together. But yeah, photosynthesis is in bioenergetics, module 4. We've already done quite a bit on respiration, but this is the first one on photosynthesis. Complete the symbol equation for photosynthesis. So what are the reactants? Because they're going to go on this side. And what are the products? Because they're going to go on this side. So photosynthesis, it needs carbon dioxide and water. They've very kindly already balanced it for us, and it doesn't matter which way round you put those. You could have put water there and CO2 there. It doesn't matter. It makes glucose, so C6H12O6 we should know is glucose, but it also makes oxygen. Nice and easy for 1 mark. One thing I should say though is make sure you make your symbols look correct. I'll just show you what I mean. Like if you do this, that is incorrect. You need capital C, capital O, and a little subscript 2. Same goes for water, capital H, subscript 2, big O. If you do this, that is incorrect, okay? It's not the correct formula. Same for oxygen. If you do this, or if you do this, that is not correct, okay? You have to do your capital O with your subscript little 2 down below. Okay, so do check that because you're going to lose an easy mark if you write the formula wrong. Figure 8 shows how the rate of photosynthesis changes with light intensity. So I always look at the axes straight away when I get a graph. I've got light intensity going up on the x-axis, and I've got the rate of photosynthesis being measured as my dependent variable on the y-axis. Which part of the graph could be represented by the equation y equals mx plus c? Well, that's the equation of a straight line. You should know, probably from GCSE maths. So we're looking for the straight line portion on the graph, which to me looks like it's here, right? Starts to curve after that, but to begin with, it's a straight line. So that's between 0 and 5 lux. Okay, so the first statement there is the correct answer because y equals mx plus c is the equation of a straight line. Okay, a student investigated the effect of colour of light. So that's what they're changing. That's their independent variable to see if it affects the results on the rate of photosynthesis. So that's what they're measuring. That's their dependent variable. In a box of leaves. On the rate of photosynthesis box. Oh, I think that's an error. Rate of photosynthesis in leaves. Figure 9 shows how the investigation was set up. Give one way the student could change the colour of light shining on the leaves. So one way. You could say use different coloured bulbs. That would be an easy way to do it. Or different coloured LED lights. As an alternative, use different coloured filters, which you'd place in front of the lamp or in front of the bulb. So you could place like a transparent filter, different coloured filters like here to change the colour of the light, or put different coloured transparent material, like a really thin plastic or tissue paper, over the lamp. Now, you do have to be quite specific. We're getting to the end of the test now. These are high-level questions. Don't just say, use a different colour filter, use transparent material. You need to say where you're going to put it. So if you're going to use a filter, you're going to place it in front of the lamp. Or if you're going to use some other kind of transparent material, you're going to say, put it over the lamp or between the lamp and the beaker. It might just be easier to say use different coloured bulbs because the others are tricky, right? Like, for example, this one, it has to be transparent. Otherwise, the light can't pass through, okay? If it's opaque, can't pass through. So if you're going to name a material, it would have to be something like tissue paper or transparent plastic or something like that. Give the independent variable and the dependent variable. Well, I think I kind of just already noted that on here, didn't I? Independent variable is the one I change. Dependent is the one I measure. So the independent variable, it said in the question, was the colour of light. Or if you're quite good at physics, you might say the wavelength of light. That would be fine as well. And then the dependent variable is what we're going to measure. I'm just going to go back a second and check what that is, because it says rate of photosynthesis, but how are we actually measuring that? Let's have a look in the table. We always put the dependent variable in the second column of the table, and we always put the independent variable in the first. So that's another trick to figure out what these variables are. So rate of photosynthesis, but we're actually measuring that by recording the time taken for 10 leaf discs to reach the surface of the solution. So I'm literally going to write what's in the table. That's what I would always do. I'm just going to go back. In the first column is what we're changing, IV. In the second column is what we're measuring to get the rate of photosynthesis, that's our DV. And I would literally just copy the exact headings from that table to make sure that I get the marks, okay? All of the air had to be removed from the leaf discs before placing them in the beaker. Suggest one reason why. So we're cutting discs from leaves. All of the air has to be removed. Now, remembering we're measuring the time taken for the 10 leaf discs to reach the surface. So this makes sense to me now. Why would we have to remove all of the air from the discs at the start of the investigation, bearing in mind we are going to be measuring the time it takes for them to float or reach the surface of the liquid. I'm probably going to say, so the discs start off at the bottom of the beaker. Also, the discs sink to the bottom of the beaker because we want to measure the time it takes for them to rise to the surface. So we don't want them to have air in them to begin with and already floating kind of halfway up. We want them to start at the bottom or sink to the bottom. What else could we say? So the discs do not float. Or you could even say to act as a control variable. If we remove all of the air from all of the discs, they'll all sink to the bottom. They'll all start at the same position. And then we can measure how long it takes for them to reach the surface of the liquid. If some of them have got some air in and some of them haven't got any air in, they'll all be at different levels in the beaker. Some might be floating up, some might be at the bottom. That's going to affect our results. That's going to affect how long it takes for them to reach the surface. So it is definitely an example of a control variable. But yeah, that makes sense. Bearing in mind the dependent variable is how long it takes for them to float to the top. We obviously want them all to start at the bottom. So it is a control variable. The leaf discs were placed in a beaker of sodium hydrogen carbonate. Explain why sodium hydrogen carbonate solution was used instead of water. So you should know from school that sodium hydrogen carbonate, or if you didn't know, think of carbonate, carbon and oxygen. It provides carbon dioxide or releases carbon dioxide. Don't just say it contains carbon dioxide. It's about providing CO2 to the leaf discs or releasing CO2 for the leaf discs because the leaf discs need CO2 for photosynthesis. Okay, you want these discs to be able to absorb the light, which is going to be all different colours or wavelengths. You want them to photosynthesize, produce oxygen, so they float up, right? Because the more oxygen they produce, the faster they will float up and reach the surface of that water or solution. But obviously to photosynthesize, they need CO2. So you're providing them with sodium hydrogen carbonate. So it releases CO2, which the leaf disc can then absorb, photosynthesize, because CO2 is needed for photosynthesis. Explain why the leaf discs moved to the surface. I think I just got ahead of myself. I think I kind of explained that in the last one. But why would the leaf discs float up? So they photosynthesize and produce oxygen, which we know, we did the word equation right at the start of this question. We know photosynthesis produces oxygen. So they're using water, they're using carbon dioxide produced by the sodium hydrogen carbonate. They're absorbing the light energy from the lamp, the different colours of light, and they will produce oxygen. Why will they float? So the oxygen, or at this point you could call it gas because you've already said oxygen for marking point 1, gets trapped in or around the leaf discs. Or you could even say the air bubbles, the oxygen bubbles get trapped in or around, or the gas bubbles get trapped in or around the leaf discs. So, just thinking this is 2 marks. So we'll push the discs up. You could explain it like that, or make the discs less dense than the solution. I hope that's making sense. If they're producing oxygen, you think about oxygen like bubbles of gas being produced, they're going to kind of build up in the leaf discs or around the leaf discs or under the leaf discs, and that's going to make the discs float up, isn't it? It's going to push the discs up or make them less dense than the solution that they're in. or move the leaves up to the surface because they've got all these little oxygen bubbles or gas or air bubbles trapped around them or in them or underneath them, you could even say. That's why they're floating to the surface. And think about this, the faster they photosynthesize, the more oxygen they produce, the faster they will therefore float to the surface. That's why we're recording the time taken for the discs to float to the surface, right? Because that can indicate the rate of photosynthesis. Okay, there are two types of chlorophyll in leaves. Figure 10 shows the percentage of different wavelengths of light that the two types of chlorophyll absorb. So we've got the two types of chlorophyll. Chlorophyll A is the dotty line, chlorophyll B is the solid line. We've got different wavelengths of light in nanometers on the x-axis, and we've got the percentage of light absorbed by the two different pigments on the y-axis. Suggest the advantage to a plant of having two types of chlorophyll. Well, you can kind of see it on the graph, right? It does say suggest, but you can kind of see it on the graph. The advantage of having those two types is to absorb more or many different colours, or we can say wavelengths, because they have used that term on the graph now, of light. Okay, because look, with a combination of both chlorophyll A and chlorophyll B, for example, chlorophyll A can absorb wavelengths of like 400, 420, 440, 450, whereas chlorophyll B can absorb more light that's around 480, 490 nanometers. Again, down this end of the spectrum, chlorophyll A is absorbing light around kind of 675 nanometers. Chlorophyll B is absorbing more light around 650 nanometers. So it's this idea that it's going to allow the plant to absorb more different wavelengths or a range of different wavelengths or many different colours of light. Now, they did also allow, which is a lot easier to get the mark, they did also allow, it helps to increase the rate of photosynthesis. You could have literally just said that and you could have still got the mark, okay? Because the more light they can absorb, the more energy they have for photosynthesis, the more glucose and oxygen they will produce. And in this practical, the more oxygen they produce, the faster they will float to the surface. Okay, the leaf discs in the investigation are green. Explain the results in table 5 for blue light and for green light. Let's just highlight that straight away so we end up talking about the right colours, because some students would talk about all three colours and you're not asked to do that. You're only asked to talk about the results in table 5 for blue light and green light. Use data from figure 10, which is repeated here, and table 6, which is repeated here. This is going to be a really hard question. It's the last question on the test. It's 4 marks. We know we've got to use all 5 pieces of information, table 5, figure 10, and table 6. We've only got to think about blue light and green light, but we do have to use data from the graph and the table. Okay, so let's have a go. Blue light and green light. Let's use this. So the colour of light, if it's blue, the range of wavelengths is 450 to 499. 450. So about here. I'm going to write that on the graph, I think. And if it's green light, it's 500 to 570. So here. Okay, so now I've done that, that's going to really help me. So I can see that blue light is absorbed way more than green light. Like there's hardly any green light being absorbed at all. So chlorophyll absorbs the most or a lot of blue light. Now you could say chlorophyll B because it's the solid line that I can see here, right? But you don't have to. You just say chlorophyll absorbs the most or it absorbs a lot of blue light. And then we could also say chlorophyll absorbs the least or very little green light. Because look down here, very, very little is being absorbed. Okay, that's nice and easy. Now we need to link that to the data in table 5. So if chlorophyll absorbs a lot of blue light, we can see the leaf discs took far less time to reach the surface, which is telling us they are photosynthesizing faster in blue light, producing more oxygen faster, which is pushing them up to the surface faster. So that makes sense. So let's try and explain this. Discs in blue light took the least amount of time to rise or float to the surface. But we have to explain why. So it's because they photosynthesized faster. Okay, so I've linked it to photosynthesis, producing more oxygen faster. That explains the results in table 5, which is what I'm doing. I'm explaining the results in table 5. If they're absorbing the most blue light, they're doing the most photosynthesis, producing the most oxygen in that short period of time, floating to the surface faster. So discs in blue light took the least amount of time to rise to the surface because they are photosynthesizing faster, because chlorophyll is able to absorb a lot of blue light. I could say the exact opposite for green, but I won't get another mark for it. So I'll just tell you what I could have said as an alternative for marking point 3. I could have said discs in green light took the most amount of time to float to the surface because they photosynthesized the slowest. I could have said that instead because I can see that in green light, the time taken for 10 leaf discs to reach the surface was the longest. And I know that's because in green light, there's hardly any light absorbed at all between those wavelengths. So there's very little light absorbed, there's far less photosynthesis, far less oxygen produced, which explains the result in table 5, that it took the longest amount of time for those leaf discs to float to the surface. Okay, let's go back to the question and see what I've not done, because I've only got 3 marks. I have explained the results in table 5, because I've done that here. I've definitely used data from the graph because I was looking at the absorption of light, wasn't I? Have I used data though? Not really used data yet, have I? Table 6, I've obviously used table 6 because I had to use table 6 to get the colour, to get the wavelength, to then look at the absorption on the graph. And again, for green, I had to use table 6 to look at green, get the wavelength, so I could identify that there was the least amount of light absorbed. So the only thing I've not really done is provided some actual numerical data. So for example, and this could be an example for marking point 4, which is for data, about 80% of blue light is absorbed. Now, if I write something like that, it's super clever because what I'm telling the examiner is I must have used table 6 because I've identified blue light. I must have correctly looked at the range of wavelengths and applied it to the graph to read off the 80%. So I've obviously used both table 6 and table or figure 10 to read off that 80%. I could do the same for green light. I could say green light, I'd have to use table 6 to find the wavelengths so I could look at the right bit of the graph. I'm looking between 500 and 570, only about less than 4%. Yeah. So I could have linked that to my first 2 bullet points. Chlorophyll absorbs the most blue light, and I could have then said, to get marking point 4, about 80% of blue light is absorbed. Or chlorophyll absorbs the least or very little green light, I could have then linked that to less than 4. It's probably about 2% of green light is absorbed. Then I'd get marking point 4 because I've used data from the graph, which I had to use table 6 to know where to look on the graph. Very hard question to finish the test with. These are very high demand. This is probably a grade 8 to 9 question, or it is a grade 8 to 9 question. Okay, we have reached the end of the paper. Let's just check. Yes, we have. We've reached the end of the paper. That was a long slog. It was a long slog for me, so I'm guessing it was a long slog for you. But hopefully, if you've got mocks coming up, or you just wanted to do some paper 1 revision, hopefully going through a whole paper like this with a variety of topics where you have to jump from one topic to another with a range of different levels of questions, a range of different command words. We saw so many graphs, we saw so many tables. This was hard, guys, so well done. I'm hoping it has been useful. Let me know if you found that useful. Let me know if you want any more of these videos. And yeah, we can go through more papers together. But do comment, let me know, like the video if you did like it. And yeah, make sure you subscribe, turn on notifications, click the little bell or whatever it is in case we do post another one, possibly for paper 2.